Good evening, one and all.
Good evening, sir.
Am I audible to you?
Yes, sir.
Today topic is plant genomics and proteomics. What is genome? Already completed in first unit. What is genome? Plant genomic and proteomics, these both are very, very important for short questions. In C8, course 8, plant genomics and proteomics both are very, very important for short questions. So let's discuss. First, what is before genomics, what is genome? And next, what is the difference between genome and genomics? And next, difference between genome, genomics, and proteomics. So in this first one, what is genome?
Genetic material present in a cell.
Genetic material present in cell.
Nucleus.
Nucleus, that is in single cell, all complete cell.
Every cell has a genome, sir.
Right, every cell is genome. So it is now the genome is complete set of genes. Complete set of genes.
Increase value.
Chromosomes, sir. Complete set of chromosomes.
But whatever it may be, where are the genes? Genes is located on the chromosomes, right?
Yes, sir.
Yes, sir.
So genome means study of complete set of genes. Here given
sir.
clearly. Please be silent. So genome is your complete set here. Complete set. Complete set. Are you able to see?
Yes, sir.
Right. Complete set of genetic information or genetic material. That is called genome. So genome, that means we have to study in the entire set of the not only single cell. Here is the complete human being made up of number of cells, right?
Yes, sir.
Just wait.
Sir, voice is not audible.
No. I'm not telling now. Why it is disappeared? Just wait. Okay, right. So entire set of genomes, it is an organism complete. It is made up of number of cells. So study of not only single cell, the entire cells, entire cell genes. So that is called a genome. So if we take single cell, this is the nucleus. It is nucleus, right? Why it is disappeared? Just wait.
और मी पंजे और क्लास है तुम्हारा पंजे
Why it is disappear immediately? What is stamp? Right. So inside cell, this is nucleus. So red color, this is nucleus. What are present in nucleus?
Chromosomes. DNA material, sorry.
First chromosomes. These are the chromosomes. In chromosomes, first cell, nucleus, inside nucleus, chromosomes, right? These are the chromosomes. The rod-shaped structure called as chromosome. What are present inside the chromosomes? Inside the chromosomes,
Genetic material.
Gene.
DNA,
DNA.
right? And what are present in DNA? Now
Genes.
genetic material present inside the DNA. That means DNA has the some active genes,
Yes, sir.
right? Do you know the DNA structure?
Double helix structure.
It is made up of DNA made up of
Nucleotide.
Keystone
yes,
particles.
nucleotide. What are those nucleotides?
Phosphate sugars and
Adenine, thymine, one and
Guanine and
nucleotides
cytosine.
are cytosine.
Adenine, thymine, guanine,
cytosine.
and cytosine. You know, the complementary bases also adenine, thymine, double bond, and thymine, adenine also double bond. Guanine,
Triple
triple bond with cytosine.
Triple bond.
Cytosine, same triple bond with guanine. So this is the arrangement of the DNA structure, right? So in genome, we have to study the entire set up to the nucleotide level. But only at the study structure of nucleotides, how they are arranged. But functionally, in function of this DNA structure and gene, the functional aspect studied in genomics. That is the difference between proteomics and genomics. Genome only entire set and study of the characters only. But we don't know how the character expressed. That is based on external environment. For example, here the two different people. This is person A and person B. Both are have the individual character, right?
Yes.
Yes, sir.
In genome only entire set of A only, entire set of B only. But we don't know how they respond to environment. That means what are the characters? The characters of the individual human beings or plants. I would like to ask a simple question. The all plants are same in a characters,
No, sir.
No, sir.
in same species. Even in same species, they are all similar characters,
No, sir.
different. Among the human being individual also different character, express their different characters. In some plants give the high yieldings, some are low yielding, some are high resistance power. Few, a few plants does not produce high quality of food, fruit and vegetables. That is belongs to their characters of the genes. In proteomics, study these are all aspects. Right? Are you clear?
Yes, sir.
Only entire set genome, but the entire set of gene respond to the environment, external environment, and express of the genes. That means gene characters. Let's see in next slide in clearly about the genomics. First, remember the first one, the entire set of the gene in organism genome, but the respond to the environment, that is called genomics. Now let me take another slide.
Sir, responding to the environment, special characters in individuals are the proteomics kada.
The proteomics also I will explain later.
Okay.
The small difference.
Okay, thank you.
Where the share? Stop share. Right. Is it clear?
Yes, sir.
Right, in genome, clear. In genomics, clearly we have to discuss about genome. Discuss about genome. Already we discussed genome. What is entire set of the, set of the genes, right? Okay, that is clear already. Genome's definition clear. So in
Yes,
genome is the part of genomics. And another part we have to discuss epistasis. What is epistasis? Epistasis means expression of character in non-homologous chromosome. Actually, what we discuss in genomes, this is the chromosome, this is another chromosome, they are the two homologous chromosome. In diploid organism, always these chromosomes arranged like this. Chromosome arranged like this. For example, these are the two chromosomes. Two chromosomes on the locus, where are the genes are present? Genes, the blue color structure gene, blue color structure gene, black color structure gene, the black color structure gene, right? In genetics, we discuss, in genetics and genome, we discuss only same locus chromosome. This is same locus. But in proteomics, but in genomics, we discussed apart from the same locus, different locus also. Different means blue and black. That is called gene interaction. In genomics, we have to discuss if you have to discuss interaction, gene interaction between non-homologous, non-locus chromosome. That means different chromosomes of genes. Here, for example, the blue color, capital T character, another blue color, small t character. Which character obtained? That means which character expressed? Hello? That means capital T tall, small, capital T tall, small t dwarf.
Yeah. Capital T is expressed.
Capital T expressed. So this is interaction only same locus, right?
Yes.
And here also, this is also different. In black color, for example, this one, red color, this one, yellow color. Sorry, yellow color. Which character obtained?
Red color.
Because this is the dominant, right?
Yes, sir.
But now the interaction non-allelics. Epistasis means interaction between non-allelic. Actually, general character expressed between the allelic. This is allelic and these two black also allelic. These two black also allelic. But in genomics, study about non-allelics, that means blue and gene. Blue and gene. So this interaction, that means non-allelic gene interaction also study in epistasis. And another one in heterosis. What is heterosis? Are you aware about this? Heterosis means, for example, here the two parental genes, one parental character. One character is maybe tall character, medium character, the tall height 6 feet, sorry 5 feet. Another character, this is a human being or plant, tall 5 plants. But is better what is height character. Here also height of the character. Height here 5 inches, height here 4 inches. Both are produced gametes. Now fertilized. As a result, formation of offspring, F1 generation. F1 generation character may be 6 feet. Or
Maybe medium also.
that means here not only medium, 6 feet or maybe 3 feet. Which means here actually,
Beyond or below, I think.
yes, any different character. When the fertilization takes place between the two individuals, one individual 5 inches, another one 4 inches, but here the different character obtained, that is different character which may be useful or harmful. So this is called as heterosis. Is it clear?
Yes, sir.
This is the plant normal production. Example, normal production. And which is also plant, it is high resistance power. High resistance power. But the next F10 generation only obtaining high resistance power more than parental gene or more than parental character. For example, here the parental plant produces, one plant produces only 100 fruits, but an offspring plant produces 200 to 300 fruits. So it is the superior character when compared with parental. That is called as heterosis.
It may happen, yes, sir.
What?
Is this happen?
Is this happened?
Normally, is this happen?
Yes, normally happened. Actually, they are mainly we get by cross-pollination, not a self-pollination. Cross-pollination means actually they are obtained mainly after we selected the desired plant. This is the crop field. This is the mango crop field. There are 100 plants are there, but in 100 plants, only please be silent. Hello. 100 plants, only the 20 plants are high yieldings. The 80 normal. 80 plants are normal. Now crossing over between high yielding plants, selected some high yielding plants and selected for normal plants. That means before initially you have to select at the desired plants. And you have to cross it between, cross-pollinated. After cross-pollinated, then F1 generation, new superficial character obtained. This character may be this character quality higher than the parental or maybe lower than the parental. Right? What happened in self-pollination? What happened in self-pollination? In self-pollination, some of the disadvantages also. Because these 20 plants,
Same characters may be transferred.
Right, high yielding, not only this character, not a superior. High yielding, not only superior. This may have disease, lack of disease resistance, easily sensitivity to virus diseases, fungal diseases.
Whatever may be parent plant genes as per they are transferred to next generation.
Yes, next generation, but not.
If it is desired means they can follow. If it is inferior means same will be transferred to next generation.
Right, transferred, but this gene altered. Not as it is transferred. While they are transferred to one generation to another generation, the gene order altered. This altered gene may produce beneficial aspect to the offspring, next offspring. Maybe produces beneficial or harmful aspects also. So but mostly the beneficial aspect. So this condition called as heterosis. Is it clear? Any doubt?
Yes, sir.
Right. Pleiotrophy. What is pleiotrophy?
Same type of parental plant maybe.
One gene here. Are you able to see this? One gene. So this gene produce character, one character only. This is the normal situation. The capital, the blue color A, this one produce only tall plant. This one produces dwarf plants.
That means that is parent plant or parent gene.
Whatever it may be, it here one gene produces one character. But some genes produces more than one characters. Here actually one gene produce one character, right? Is it clear? Just wait, let me complete. Do you know transcription, translation?
Yes, sir. Protein synthesis has two steps. One is transcription, other is translation.
Right. For example, here, what happened here? Not. I'm telling you, just listen. In a DNA sequence, adenine, guanine, cytosine present. Adenine, guanine, cytosine, some other nitrogen bases are present. But during transcription process, from the adenine, uracil produced, right? Hello?
Yes, sir.
To disappear. ఎందుకు ఇట్లా పోతుంది? Right. This is the DNA sequence, okay? From the DNA, adenine, uracil, thymine, adenine, guanine, cytosine, cytosine, guanine, right? This is mRNA. Are you follow me?
Yes, sir.
RNA, this is DNA. On the RNA stand, protein prepared. Here, how many proteins are formed? How many proteins are formed here?
Only one.
One protein, right? These
Yes,
proteins are enzymes. Already told you in the protein synthesis or
yes.
gene regulation and gene expression.
Yes, sir.
Now these enzymes express the characters. Express the characters. Now here, how many character? One character for one gene, right? Hello?
Yes, sir.
Right. What if the changes, the loss of thymine, the same genetic order, already after formation, one protein, the same order lost the thymine. As a result, what about the sequence of RNA? Same UA, CG, or it is altered? Is it altered or same?
Altered.
Altered. Tell me the sequence.
ULG, UCG.
U, U, UC,
G.
UCG. Now, what about the protein? Is it produce the same protein or different protein?
Different protein.
Protein blue, and this protein character, red character or blue character?
Blue character, that is other character.
Right. And now this time changes C, last one.
Sign.
Or actually it is duplicate, C duplicated. Now the sequence of RNA,
Changes.
U, A, C, G and
G.
G. But the previous, these are nucleotide same. But as added the last one, automatically the protein synthesis also changed order. Now the protein also different protein formed and produces the different character.
Characters.
Also change. But here, how many genes here? This is gene.
Single gene.
This is gene. Gene single. Gene single. But how many character produced? Different various characters are produced.
2 characters.
Right. I am giving just example. Like that, number of characters are formed. Number of characters are formed. This is called as pleiotrophism. This is called pleiotrophy. So study of pleiotrophy, study of heterosis, study of epistasis, study of genome. These are all comes under the genomics. Right? Clear?
Yes, sir.
Yes, sir.
Right. Genome and genomics. Genome already we discussed in first unit. Entire set, and it is composition of nitrogen bases. That's it. And genomic especially not here. Here remember the genomics. In genomics, study about only active genes. Study only active genes. Means actually, for example, the 100% of genes are present in human being. But out of the 100, only 2 to 3, 4% active gene. What about rest of the gene? The rest of the gene called as junk gene. Means non-coding. Gene present, but does not code for any RNA, does not code for any protein. So these are called as junk genes. So only but here active genes only 2 to 4%. And another one is in all individuals, almost 98% genomes are similar character, similar. But the variation only 1.2%. 1.2%. In genomics, especially the study about this special 1 to 2% only. Not all. 1 to 2%. And what is the difference between 1% to another person? 1% to another person? And the behavior character of the person and behavior character of this person, the variation between among the individuals. So these are all we have to study in genomics, right? Is it clear?
Yes, sir.
The same way, plant genomics. Plant genomics, the study of genomics of plant. The same study, but inside the plant, that's it. Now proteomics. Proteomics means the study of protein, simple. You know already how the proteins are formed? How the proteins are formed?
From DNA.
From DNA.
DNA.
By
Yes, sir.
transcription translation process.
Yes, sir.
By transcription translation process. Let's see difference between genomics and proteomics. Genomics and proteomics. Right here, genomics. Genomics, proteomics, proteomics. Genomics here the stable. This is stable. Same in individual. In individual cells also same. For example, here taken one organism. Any organism made up of how many cells? It is uncountable cells, right? Millions of cells. But what about the gene order in each and every cell? Same or different? Hello?
Different.
Same set of chromosomes.
Same set of chromosome, because remember the organism developed from the only from single cell. The single cell called as zygote, right?
Yes, sir.
Zygote. For example, here the 46 chromosome initially. Single cell with 46 chromosomes, same chromosomes. Later, by mitotic, it is converted into two daughter cell. In two daughter cells, here also 46 and here also 46. Is there any changes?
No, no.
Like that, uncountable cells are formed. Group of cells called as embryonic cell. After formation embryonic cell, these are differentiated into parts, root, stem, and leaves. So in each leaf and root and stem branches, each cell contains the same genetic material. What about proteomics? After formation protein, the proteomics also same or different? Proteomics also same or different? Already told you in third in it.
Different proteins are
Proteomics,
formed,
that
right?
means the proteomics, remember, first of all, the proteomics means the complete set of proteins of an organism. Study of complete set of proteins of an organism. That is called proteomics. Complete set of genes of an organism called as genomics. Right?
Proteomics is individual.
Right, but genomics is similar. In individual cells, all cells are made up of same type of genes. What about proteins? The proteins also similar or different?
Different.
Already you know the formation of protein from the gene, right? from the gene to proteins formed.
Protein.
Okay.
Same proteins.
Do you know gene expression? Already told you gene expression. Hello?
Maybe same, sir.
What is gene
But
expression?
every gene expresses
Expression of characters.
Have you heard the word luxury genes? Luxury genes word already explained in offline class.
Yes, sir. Yes.
What is luxury gene?
కొన్ని యాక్టివ్ ఉంటాయి, కొన్ని ఇన్యాక్టివ్ ఉంటాయి జీన్స్.
Give me example.
Not example.
I have given 2 example on the class. One example. Please tell me one example for luxury genes.
Example.
In an organism, this is the cell and this is also cell. Right? This is leaf cell. This is flower cell. Flower cell. Right? What about genes in this all? Genes.
Same, sir.
Flower also same gene. Right?
But proteins will be different for both of them.
Right. In what happened in gene expression, some genes active in leaf, some genes active in flower. Here only the active genes in leaf, mainly chlorophyll pigmented. Chlorophyll pigment. In flower, color pigment. Color pigment.
Chromatophores, etc.
Right. Color pigment, mainly keratin. Or some other, any pigment, any coloring pigment active. But the coloring gene, does it have the coloring pigment in leaf? Coloring pigment gene?
No.
I'm asking coloring pigment gene.
Gene will be present but will not be expressed in.
Right, gene present. Here chlorophyll, here also chlorophyll gene present. But chlorophyll gene not enter into the transcription process. Here pigmented gene not enter into the transcription process. Right?
Yes, sir.
But here only chlorophyll pigment enter transcription, translation, and chlorophyll protein form. Here same pigment protein form. And another example in human beings, the skin cells and blood cell. Skin cell gene same, blood cell gene same. But what about proteins of the skin cell and blood cell? The both are same pigment, same proteins or different protein?
Different.
Hemoglobin protein present in skin?
No.
Melanin present in skin, hemoglobin present in blood.
Right. So here genomics, all genes are similar. Unity, no changes. But in proteomics, different proteins are formed based on the cell structure or cell function. Based on the cell function. So that is called proteomics. Right?
Yes, sir.
Clear?
Yes, sir.
So remember, genomics, entire set of the gene of an organism, and study entire set, and study of interaction of genes, and study of another one, pleiotropic, another one, heterosis. These are all in genomics, right? What about proteomics?
Only
Proteomics,
about
simple
protein.
definition.
Study of complete set of proteins of an organism is called proteomics.
Right. Another difference is in an organism, number of cells are present, but in protein composition, different in different cells, but not all are same. But in genomics, the all cells are made up of the same genetic material, same component. Whereas proteomics, the different material, different protein materials are formed. That is based on requirement of an organism. Right? Clear?
Yes, sir.
Now, genomic research. Genomic research, some are the scientists given in page number 254. Walter Fairs and Frederick Sanger. And the gene introduced by Winkler. These are involved in genomic research. The first actually the genomic research started by bacteriophage genomes. Bacteriophages were historically used to define gene structure and gene regulation. The first genome sequence studied in bacteriophage in 1879. Next, bacteria genomes. What is bacteriophage?
Viruses which attack bacteria.
Right. Bacteria. Next one, bacteria. In 2007, about 577 bacteria species studied, such as Haemophilus influenza. The best example, Haemophilus influenza. And apart from that, 24 cyanobacteria cyanobacterial genome also studied. And some eukaryotic genes, such as example, yeast and fungi, and Arabidopsis, Drosophila. These are all studied. But mainly, the best convenient plant is Arabidopsis. And Human Genome Project. What we study in Human Genome Project?
All chromosomes, genome sequence.
Genome sequences of all individuals, and also compare the difference. There are 3 people are there, A and B and C. Why A people behave like that, and B people behave like that, and C people behave like that? What are the reasons behind it? They have to find out in human genomics, Human Genome Project.
Variable repeat sequences.
Right. Actually, this project started, I think, 2001. It completed by 2007. All individuals, about 2000 bases, nitrogen bases are there. That is common.
20,000.
Right, 20,000. Right, 20,000. Human Genome Project, 13-year international project coordinated by United States Department of Energy and National Institution of Health. What are the goals of this project? The first one, identify all the approximate genes which are 2000 to 2500 in human DNA. Especially in these genes, not for junk genes, only active genes. They are separated in this project, separated active genes, only 2% gene, and analyze the 2% gene among the individuals and find out the reason of their individual characters. And next, determine the sequence of the 3 billion chemical base pairs of the human DNA. 3 billion genes, 20,000 to 25,000. Chemical bases, nitrogen bases, 3 billion. And this information stored in database, computer information, that is called bioinformatics. Apart from the human genome project, after analysis the genetical order, gene order, these order stored in computer bases. This is called bioinformatics. And next one, improve the tools for data analysis. That is best way. Find out the best way to analyze the genes, genetic order. Next, address the ethical and legal and social issues. And human genome contains 3.2 billion nucleotide base pair already repeated. Don't need. Right, to about 2%, okay. Right. Not in this project, not only about genomes, and also study about proteomics also. And functions are unknown for more than 50% discovered genes, junk genes. They're unknown function. Why they are unknown? They cannot code for any gene, any character.
Inactive.
Not code for any RNA, not code for any protein.
Protein synthesis.
That's why these are useless. Right?
Silent genes, can we call them, sir?
What?
Silent genes, they are, sir.
Yeah, silent genes.
Yes.
Another name, silent genes, junk genes. Right. I've given about Arabidopsis, but no need to explain here. But here only the examination point of view, just write about proteomics or genomics. That's enough. Right, species and some important crops there, genomes. No need. Proteomic research, practical genomics, proteomics. Right. This is only short answer question. And next one, let me complete first. Of course, here Arabidopsis not only for theory, not important for theory exam, but is important in competitive level. Arabidopsis thaliana, first high profile model plant. It is being used around 800 years. But in 1996, United States and Japan, Europe established the Arabidopsis genome. It is completed year in 2000. That is the Arabidopsis genome was fully sequenced in 2000 year. And the total number of gene was just over 25,000. Arabidopsis has more duplicated genes and multi-gene families than non-plant eukaryotics, mainly because of the effect polyploidy, which rare in animal kingdom, right? So in this plant, right, typing. Right? What do they find out in this plant? Multiple genes and duplicated genes. They find out multiple genes, duplicated genes in Arabidopsis plant. And any explanation here? The plant genomic research no need. So apart from the Arabidopsis, some other plants also studied genomics and proteomics, such as eggplant, tomato plant. Eggplant means nothing but that is the brinjal plant. Studied genomics, proteomics of eggplant and tomato, potato and pepper plants. And in 2003, several higher plants and crops like rice, genomes and sorghum, maize also studied. And these are studied and stored inside the computer database for further use. And some are the plants given, page number 259, table 18.1, some important crops and their genomes. Oil seeds, oil rape seeds, cereals, legumes, and some Solanaceae family. But they find out some genome size variation among the individuals. Arabidopsis thaliana 1.3, Brassica nipus 1.2, Oryza sativa 4.2, Hordeum vulgare 4.8, Triticum aestivum 1.6, Giames 2.5, Pisum sativum 4.1, Glassine marks 1.1, Solanum tuberosum, Lycopersicon. So there are different genome size. Apart from the genome size, actually not given, and also they find out the special character of these individuals. Right? Are you follow? Page number 259.
Yes, sir.
Yes, sir.
Right, EST, expressed sequence tags, no need. Expressed sequence tags, no need. This one, but it is
Sir,
not
ఇక్కడ
clear.
అదర్ ప్లాంట్ జీనోమిక్ రీసెర్చ్ కూడా ఎగ్జామ్ పాయింట్ ఆఫ్ వ్యూ రాదు కదా?
Other plants mean?
జీనోమిక్ రీసెర్చ్ ఎగ్జామ్ పాయింట్ ఆఫ్ వ్యూ లో ఇస్తారా?
Competitive.
Competitive.
But in theory, only short question, one
Genomics
word,
and-
that is genomics, enough.
Okay.
Proteomics, enough. And
Okay.
apart from that, another important one is genomics, proteomics, and next topic. And the proteomics application, genomics application, let's see.
ప్రాక్టికల్ అప్లికేషన్స్ ఉంది.
Right. Human genome projects also not given here. But if they ask about human genome project, you have to write about both genomics and proteomics.
Okay, sir.
And aims. Aims of the page number 256, human genomics. When they started, when they completed, which university organized to conduct, which university conduct to this project, and main goals, aims, goals of this project. Right?
Okay.
And just write about some definition about proteomics and genomics before that. Right? Some of the applications of genomics and proteomics. Last, after write about definition and concept of genomics and proteomics in end with some of the application.
Okay, sir.
The main application, identifying the genes and gene products in the organism that lead to disease in human will lead to the development of treatment for the diseases. If you study the gene and gene character, for example, here some of the scientists find out a diabetic gene. Why in diabetic patients, diabetic character expressed? Which gene responsible for that? If we find out,
insulin gene is dysfunctional.
right. If we find out, is there solution to stop this activity?
Therapy can be done, sir.
Therapy, that is called gene therapy.
Gene therapy.
So the best application of this project of the proteomics and genomics is gene therapy. Right? That's enough. And why these are the proteins? No need. So next one, plant metabolomics. Right. This is also short question, short answer. Plant metabolics.
Sir, plant
What?
genomics and proteomics నుంచి షార్ట్ ఆన్సరే వస్తుందా సార్?
అవునండి.
ఓకే.
This way. Right. Plant metabolomics means gene from the gene RNA, from RNA protein, right? This protein enzymes involved in biochemical reactions. Biochemical reaction, as a result, formation of product, chemical product. Chemical products. The study of these chemical products called as plant metabolomics. What are the chemicals? What are the chemicals?
Carbohydrates, proteins,
And
fats.
Hi, sir.
mainly not a primary products, mainly the study of secondary products. What are the secondary products?
Terpenoids, flavonoids, etc. sir, in plants.
Resins, tannins.
Right. So if we take the photosynthesis first, in photosynthesis, what happened? Photosynthesis enzymes are involved to preparation of food material, right? To preparation of food material, photosynthesis enzymes are involved. How the formation of photosynthesis enzyme? By translation and transcription, as a result, proteins are formed.
ఆమె ఏం చేస్తుంది చిక్కి?
Right. So these photosynthetic enzymes involved in light reaction And dot reaction, as a result, finally formation food. So earlierly we study about genomics, and next topic study about proteomics. But now study about after end product of this gene and protein, gene activity and protein functional activity. And after formation, this some of the proteins, biochemical activity of this biochemical activity, food material formed. After utilize, this is primary food. As you said, carbohydrates primary, and also proteins, also protein formation, also primary. And next, fat formation primary. But after formation, this primary product, these are utilized for activities, organism activities.
Anabolic.
After,
metabolic,
right,
yeah.
these carbohydrate, protein, these are after utilized. What about the waste material? That is called as secondary product. These are primary product. After utilized, from this utilized product, secondary material formed. Secondary product formed. This secondary product, actually in animals, these are stored or excretory out.
Treated out.
Excretion, it's involved in excretion process and leaved out. And very few of the chemicals in the form of nitrogen stored, but very rare. But what about in plant? Is there any excretory system in plant body?
No, in the form of seed కానివ్వండి అట్లా.
Right, in plant parts, in plant parts, maybe root and stem and leaf, they are stored in form of nitrogen, such as alkalides, in the form of alkalides, and form of terpenoids. What are given in our book? Alkalides, not given, okay, but you have to write the alkalides, terpenoids,
Resins, gum, mucilates.
and phenolic.
Latex.
Resins also, phenolic.
Resins, latex.
Resins, latex, they are all.
Tannins also.
Tannins also. They are all. So the plant metabolics only study of chemicals. How the formation of chemical after formation, primary products after utilizing these secondary products are formed in the form of alkalides, terpenoids, and phenolics. Terpenes are liquid synthesized from when these terpenes are formed. These terpenes are formed by acetyl coenzyme A, synthesis of acetyl coenzyme A, or from intermediate of glycolysis. Actually, lipid synthesis not complete. Okay, fourth or second year, kada? Right? But in first year, lipid synthesis there. In lipid synthesis, these lipids are synthesized from the acetyl coenzyme A. That means these terpenes mainly formation intermediate of Krebs cycle and glycolysis, both Krebs cycles and glycolysis. And these terpenes, terpenoids constitute largest class of secondary product. Already told you, these are the secondary product. These are water soluble, common in biosynthetic origin from the acetyl coenzyme A glycolytic intermediates. Only one word enough. Terpenoids, which are formed in intermediate product of Krebs and glycolysis. Enough, one word enough. Don't write this all. And what about phenolic compounds? Phenolic compounds are aromatic substance. What is aromatic? Hello?
Ringed carbon compound. Having aroma.
Scented smell.
Substances.
Scented smell.
Smell.
Right. So these are formed cyclic acid pathway or malonic acid pathway. These two pathway not in our syllabus. But remember, cyclic acid pathway and malonic acid pathway. During this pathway, these phenolic compounds are formed. The plant phenolics are chemically heterogeneous groups. This is no need. That one word enough. These are organic solvent. Terpenoids water soluble. These are organic solvents, enough. And defense compound against observing UV radiation, harmful UV radiation, reducing the growth nearby competing plants. These are in attracting pollinators. The function of phelonic compounds, attracting pollinators, pollinating insect, and observing harmful UV radiation, and reduce growth of nearby competing plants. That means herbs. It is act as a herbicide, right? And nitrogen bases, already some of the alkaloids are there. What is the main purpose of alkaloids? Why they are used? The nitrogen bases alkaloid mainly stored inside the seeds and for leaf or fruit.
Medicinal values.
Medicinal value. For drug preparation, drug discovery. These phenolics for drug discovery. Right? Any there? Analytical technology, secondary. Right. So now, okay, the study of chemicals, that is secondary metabolites. Study of secondary metabolites called as plant metabolomics. How they are extracted from the plant? What are the method to extract this secondary metabolism? Here they given some methods: gas chromatography, high performance liquid chromatography, HPLC, or mass spectrophotometry, and nuclear magnetic resonance, NMR, Fourier transform infrared, FTIR. These are the method from extraction of this compound from the plant body. Is it clear? So in plant metabolomics, you have to study first definition, how they are formed, process or formation. If it is present inside the plant, how to find out, how to extract first. Extraction methods. And next, metabolic system biology. What is it? It is right. It's the bogus. Now let's jump in summary. No need. Right. So these three compulsory question. And another last unit also, intellectual property rights. Intellectual property rights. Only these are for short answer. Proteomics, genomics, plant, metabolomics. Right. What is intellectual property? What is intellectual property? What is intellectual property, right?
Intelligence.
What is intelligent?
Patents will be granted for a particular
right.
research.
Patent. For example, if you find out the new thing, new discovery, the whole and sole rights for you only.
Yes, sir.
Right? In intellectual property rights, history, no need. They are given IP. First definition in introduction given, an intellectual property right is legal concept that confer the right to owner.
And
It depends on who is owner and creator of the work, creator of the new work, novel work, write down, creators of the novel work for their intellectual creativity. Write down novel work, creators of the novel work.
Novel, sir. In
yes, new.
Okay.
And India renowned scientist discover no need, scientist report molecular state no need, Raman effect no need, Geography, sorry, no, history. Just add this history. Actually, history also no need, but exam point of view, no need. But if you curiosity to know about this, just read it. 500 BC, I think King James, no need. These are the waste material. Branches of intellectual property. Industrial property, invention of patent, trademarks, industrial designs and wrong but చూసుకోండి. Geographical indication source, copyrights. First one, industrial property, invention, patent. Patent is a legal monopoly. This is first one only in research areas. Only in research area. In research area, if you find out new thing, new concept, on this new concept, you have to file a patent. After filing a patent, it would be granted later. Now the issue the license. That is the last stage. Trademarks given the goods, materials. Industrial designs, this feature, shape, configuration, pattern, ornament, compositions, lines, color, combination. This is only the factory development. No need. Industrial design, no need. Geographical India, this is not part of our subject. Copyright, this is common. Copyright. Why we give copyright? Books, writers.
Duplicate should not be made by others.
No one should copy.
Books, manaku notes, books రాస్తారు కదా writers.
Authenticity can be made.
వేరే వాళ్ళు నాది అని చెప్పకుండా.
రైట్ అంటే వేరియస్ బ్రాంచెస్ కి ఏమేమి ఉన్నాయి ఇచ్చిండు అంతే ఇక్కడ. పేటెంట్ దేనికి, ట్రేడ్ మార్క్స్ దేనికి, ఇండస్ట్రియల్ డిజైన్ అవసరం లేదు. లాస్ట్ కాపీ రైట్ అంతే. రైట్. ఇప్పుడు మీకు దీంట్లో ఇంకేం ఇంటెలెక్చువల్ ప్రాపర్టీ ఆర్గనైజేషన్. ఈ ఆర్గనైజేషన్స్ ఇంకొంచెం నెట్లో నుంచి కొన్ని తీసుకోండి. హలో, ఇంటెలెక్చువల్ ప్రాపర్టీ ఆర్గనైజేషన్స్ ఇక్కడ ఉన్నాయి కదా, రీసెంట్ ఉన్నాయి, నెట్ లో ఉంటాయి చూడండి.
ఓకే సార్.
రీసెంట్ రాయండి, ఇన్ని అవసరం లేదు. ఇంటెలెక్చువల్ ప్రాపర్టీ రైట్ స్టేటస్ ఇన్ ఇండియా. ఇంటెలెక్చువల్ ప్రాపర్టీ స్టేటస్ ఇన్ ఇండియా ఉంది కదండి, అది ఇప్పుడు ఈ బుక్ లో కాకుండా మీరు గూగుల్లో సెర్చ్ చేయండి. చేసి అదే ఇన్ఫర్మేషన్ తీసుకోండి, దాన్నే మీరు ఎగ్జామ్ కూడా రాయాల్సి వస్తుంది.
ఓకే సార్.
ఇదంతా వద్దండి, ఇదంతా ఏమేమి ఇచ్చింది ఇక్కడ, ఇదంతా సిబిడి, బయోసేఫ్టీ, ఇండియా ఇన్స్టిట్యూషన్ డీల్ విత్ బయోసేఫ్టీ, ఇంత పెద్దగా అవసరం లేదు. జస్ట్ వెయిట్ అండి. Intellectual property rights. మీకు సపరేట్ వెబ్సైట్ వస్తుందండి, ఇంటెలెక్చువల్ ప్రాపర్టీ సైట్ వెబ్సైట్. దీంట్లో మనం ఏం తీసుకుంటాము, డస్ Intellectual property rights, what is the status in India? The first patent law was introduced in India 1856. ఇట్లా ఇది ఒక కరెక్ట్ గా ఆర్డర్ ఉందండి. ఇది రాసుకోండి ఇట్లా.
1856. పేజ్ నెంబర్.
పేజ్ నెంబర్ ఇక్కడ దాంట్లో లేదు. షార్ట్ కట్ రాసుకోండి ఒక 5 minutes లో, ఇంతే, ఎక్కువ రాయకండి. ద ఫస్ట్ పేటెంట్ లా వాజ్ ఇంట్రడ్యూస్డ్ ఇన్ ఇండియా 1856 విచ్ వాస్ ఫాలోడ్ బై ద నేను షేర్ చేస్తానండి ఇక నేను పంపిస్తాను. ఛాలెంజెస్ ఐపీఆర్, ఫ్యూచర్ ఫార్వర్డ్ గవర్నమెంట్ ఇనిషియేటివ్స్ కొన్ని కట్ పేస్ట్ చేసి నేను చేంజ్ చేసి పంపిస్తాను. ఇది దీనికి ఎక్కువ ఏం టైం లేదు. రైట్ అండి, ఇంతవరకు క్లియరా?
సార్.
What are the remaining topics in C8? First blank, blank 1, molecular biology 1, genome completed, genome organization in higher plants completed. Chloroplast, mitochondrial genome, structural organization in eukaryote, gene expression, eukaryote, regulation of gene. First blank completed, right? Second block restriction not completed, modified engine not completed. Second block not completed. Third block completed.
అయిపోయింది.
Fourth block also completed.
లాస్ట్ లో ఇవి ఉన్నాయి కదా సార్, ఇది అవసరం లేదా? ఇంటలెక్చువల్ ప్రాపర్టీస్ రైట్స్ అండ్ కన్వెన్షన్.
I will send the notes.
Biodiversity.
Biosafety also. Actually in our book given a lot of information.
Okay sir.
ఇంత అవసరం లేదు. అంతే కొంచెం జిగ్జాగ్ గా కన్ఫ్యూజన్ గా ఇచ్చిండు. అంత ఒక ప్రాపర్ వేలో లేకుండా. ఐ విల్ అరేంజ్ ద కంప్లీట్ సెటప్.
ఓకే సార్.
మొత్తం ఒక చిన్న సెటప్ మంచిగా అంటే నీట్ గా రాసేసి మీకు పంపిస్తాయి. ఇది ఇంటలెక్చువల్ ప్రాపర్టీ రైట్స్ ఒకటి, బయోసేఫ్టీ ఇంపార్టెంట్ కాదు కానీ చూసుకోండి, ఇది కూడా చదవండి. అంటే మనకు ఒకటే బ్లాక్ ఉందిగా. మాలిక్యులర్ బయాలజీ, రైట్? అసైన్మెంట్స్ కంప్లీట్ చేశారా?
లేదు సార్, స్టార్ట్ చేయాలి.
ఓకే
సార్, ఒక కన్సర్న్, మేడం తో మాట్లాడమన్నారు కదా ఎగ్జామ్స్ కి. మేము అప్పుడు అడిగాం కదా ఎగ్జామ్స్ ఆల్టర్నేట్ డేస్.
అవన్నీ ఇప్పుడు కాదండి, క్లాస్ లో కాదు, తర్వాత చెప్పండి, మాట్లాడదాం. మరి ఇక నెక్స్ట్ ఈరోజు ఫ్రైడే కదా, సారీ వెడ్నెస్డే కదా. ఫ్రైడే నుంచి వేరే సార్ ఒక 2 days తీసుకుంటాడండి మీ క్లాస్ లోని. క్రాంతి సార్ తీసుకుంటాడు. నేను అవైలబుల్ ఉంటలేను, క్రాంతి సార్ తీసుకుంటాడు. ఉండండి, ఓకేనా?
ఓకే సార్.
రైట్ అండి.
థ్యాంక్ యూ సార్.
రైట్.